Hello, I'm Dr. Hoon.
And I'm Dr. Eric Dent, and we're part of the Neuroscience Training Program at the University of Wisconsin-Madison.
We want to give students the opportunity to interact with neuroscientists like us. So on Thursday, October 11th, the Neuroscience Training Program is hosting an Ask Me Anything live chat session on Reddit from 9 a.m. to 4 p.m.
Use the link in the description of this video to join the live chat and see what students around Wisconsin are asking, and ask questions of your own. My lab is interested in something called the cytoskeleton. Cyto means cell, and skeleton means just like your skeleton of your body. Here you can see neurons cut from a slice of brain. We've labeled all of these neurons. Here are the cell bodies here are the dendrites up here, and the axons down here. This is similar to what you would see with your pipe cleaner neurons. They would be positioned just like this with the dendrites going up and the axons going down here. We're very interested in neuronal development, and you can see here a neuron that we've taken out of a brain and plated in culture and how it grows over time. So this is the time in several hours, but you can see how dynamic this process is. We're also interested in how this process grows out and understanding the cytoskeletal mechanisms around that. And you can see here we've labeled just two proteins, microtubules in here in green and actin in red. And during this movie you can see how dynamic these microtubules are growing down the axons here or into the dendrites here. A lot of people think that the cytoskeleton, just like your bones, are actually stable, like train tracks, but these are actually very dynamic, more similar to the stairs in Harry Potter that move over time. We're interested in looking at development because we feel that understanding development will put us in a much better position to understand when things go wrong during disease states.
So my lab studies the retina. The retina is a layer of tissue that lines the back of our eye and contains all the receptors of the visual system, allowing us to see the world around us. We study connections between retinal neurons in development and also in disease, with the hope of finding out what goes wrong with these connections during disease conditions. So what are these connections? Retinal neurons, like all neurons in the brain, they connect at junctions called synapses. So synapses are the point of communication between two neurons, typically the axon terminal of a presynaptic neuron with the dendrite of a postsynaptic neuron. Now the presynaptic neuron contains these chemicals called neurotransmitters, which are then released across the synaptic cleft to activate receptors on the postsynaptic neuron. Thereby the signal passes from the presynaptic neuron to the postsynaptic neuron. But studying these connections is incredibly hard. They're very densely packed. The distance between the presynaptic and a postsynaptic neuron is really tiny, about 1,000 times less than the diameter of your own hair. So it's complicated, but it's interesting, and we look at these connections in the retina. We want to also know what's happening to these connections in disease to look at finding ways for therapies in the disease conditions.
We hope you enjoyed learning about what we do, but this is just a tiny glimpse of the research done here. Neuroscientists at the University of Wisconsin-Madison study everything from memory to sleep to concussions.
All different kinds of neuroscientists will be answering questions live all day at our Ask Me Anything event, so be sure to join in Thursday, October 11th. To get us started, here are the answers to a few questions. So Dr. Dent, what is the coolest part of your job?
I think the coolest part is knowing that every day I walk into the lab, I'm seeing something that nobody in the world has ever seen before. Discovering new parts of the nervous system is really fascinating and really interesting. At what point, Dr. Moon, in your career did you decide to become a neuroscientist?
I think that was around the time when I was an undergrad and learning about the nervous system and the complexity of the nervous system and how diseases of the nervous system are so difficult to come up, to treat. And that just got me thinking that the neurons in the brain have this one-shot chance of making the right connection. And that just led me to learn more about these connections in disease and become a neuroscientist.
Neuroscience Training Program members encourage you to ask me anything on October 11th. We look forward to answering many more of your great questions. We'll see you there.